Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this session is meant for 29th June 2021. And in this session we are going to deal with chapter 2, sexual reproduction in flowering plants. Yesterday I had introduced some technical terms to you, and today we are going to talk in detail about particularly Androecium, male reproductive system. So basically the reproductive part in plants is the flower. So without flower there is no sexual reproduction. All flowering plants, we call them angiosperms. All angiosperms show sexual reproduction because they are flowering plants. So flowers are the sites of sexual reproduction. And before actual fertilization, the flower has to prepare itself. So this is called pre-fertilization structures. So several hormonal and structural changes take place that result in differentiation and development of floral primordium. So basically this floral primordium refers to inflorescence, that is they bear floral buds and the buds develop into flowers. So floral buds will become flowers. So typical structure of a flower, of course already we said yesterday, a flower consists of 4 whorls. The green sepals will be there outside. When the flower is in the form of bud stage, it gives protection, and we said calyx, and then the colorful, very fragrant petals will be there, and this group of petals is corolla. So calyx, corolla. Then why flower is called reproductive structure? Because of the presence of 2 more whorls. One is Androecium and the next one is Gynoecium. Androecium consists of male reproductive organ and Gynoecium consists of female reproductive organ. And how do they develop? Now it is the Androecium, as I said, is the male reproductive part of the flower. It consists of a whole of stamens, you can see, and their number and length are variable in different species. As I said, the number of stamens will vary. So basically you got the stamen, and the stamen will have 2 parts, one bulged portion that is anther and one tube-like structure called filament. And when the anther becomes matured, it breaks open. We can see these structures called sporangium. So there is connective which makes the structure into bilobed. The anther becomes bilobed because of connective. And further, as we said, the stamen has got 2 parts, one anther part, bulged portion, and another one is filament. And inside there is microsporangium inside the anther. And further, when we take this filament, as we said, filament is a stalk that bears the anther. And when we take the TS, transverse section of the filamental stalk, we can see the presence of in the anther pollen sacs. And in the pollen sacs there will be presence of pollen grains. This is on maturity. And further, dehiscence takes place. The anther becomes mature and breaks open, and the pollen grains will be released into the atmosphere. So basically it is the filament that bears the anther. So filament is a long and slender stalk. Its proximal end is attached to thalamus, as you are seeing in the picture, and the other end, that is the other end bears the anther. And further, the Androecium, which is the male reproductive organ, bears anther. So anther is terminal and typically bilobed, means lobe means room, 2 structures. As you are seeing, it breaks vertically, anther breaks vertically and forms 2 rooms, so bilobed. Each lobe has 2 theca or thecae. So we say dithecous, as you are seeing. Further there will be 2 rooms. Each lobe has got two rooms. So basically it is bilobed and it has got 4 rooms. Often a longitudinal groove runs lengthwise separating the theca, as you are seeing here. So there is a line which is vertical. And when we take the transverse section of anther, this is what it appears like. The anther is tetragonal structure. Tetra means 4. 4 rooms will be there. And it consists of 4 microsporangia located at the corners. Look at that, microsporangia. And that in each lobe there will be 2. The microsporangia develop to pollen sacs, as you are seeing here. Pollen sac formation will be there, microsporangia. They extend longitudinally, okay, as you are seeing, they extend longitudinally and through the length of the anther and are packed with pollen grains. So on maturity, the sporangia will be packed with pollen grains. Since it is packed with pollen grains, we say pollen sacs. And then when we talk of the microsporangium, further when we go interior, a typical microsporangium is near circular in outline. It appears like a round structure and it is surrounded by 4 wall layers, as you are seeing. First one is epididymis, external most, outermost one, external one. Second one is endothecum, as you are seeing, the inner layer. Third one is the middle layers. Further you can see the middle layers inside. Then the fourth one is the tapetal layer, and after the tapetal layers you can see MMCs, microspore mother cells. That means further when we go, when we take the section in the Androecium and Microsporangium, we see this is how the Microsporangium will be there. Basically epididymis is 1 cell thick and protective in function. You can see here in the diagram, it is 1 wall, 1 thick, 1 cell thick, and it is protective. Endo means inside, thecium means layer. Endothecium is single layered again. Cells have cellulose thickening with a little pectin or lignin. In some cases, it helps in dehiscence, means breaking. Middle layers, which we are talking of, they are 1 to 6 layers. They degenerate during maturity of an anther. They are just for protection. On maturity, they degenerate. And most important is tapetum. This is one more question again. Tapetal cells are nutritive. They are for nutrition. By these cells, the ubic bodies are produced which help in the ornamentation of microspore wall. A compound, sporopollenin, is secreted by ubic bodies which is deposited in the exine of microspore wall. Okay, so the outer 3 layers give protection, that is epididymis, endothecum, middle layers, and help in dehiscence of anther to release the pollen. And the tapetum, as we talked about it, innermost layer nourishes the developing pollen grains. Cells of the tapetum contain dense cytoplasm and generally have more than 1 nucleus. You see, it is multinucleate. Tapetal layers are multinucleate, as you are seeing. And in a young anther, each microsporangium has sporogenous tissue at the center. So there is a sporogenous tissue at the center. And this, it consists of compactly arranged sporogenous cells. These are diploid in nature, 2n. Okay, you can see the tapetal layers, which are after the tapetal layers, you can see the centrally sporogenous tissue. Further, how this microsporogenesis takes place. What is microsporogenesis? I said it is the process of formation of microspores. Okay, and you can see here, as the anther develops, each sporogenous cell, it is called MMC or PMC, yesterday I said, microspore mother cell or pollen mother cell, undergoes meiosis, I said meiotic division, to form microspore tetrads. That means 4 will be there, 4 cells will be there. So that's why tetrad. So microspores are arranged in a cluster of 4 cells. The formation of microspores from a pollen mother cell, PMC, through meiosis is called microsporogenesis. You can see the same thing. The PMC, pollen mother cell, is undergoing meiosis and forming tetrads, and these tetrads become pollen grains or microspores. Since microspores are being formed from the PMC by undergoing meiosis, it is called microsporogenesis. And further, the microsporogenesis, as the anthers mature and dehydrate means there is no water, moisture will not be there. The microspores dissociate from each other and develop into pollen grains. So the tetrads will be breaking apart and each one becomes pollen grain. Now each microsporangium contains thousands of pollen grains like this, and they are released with the dehiscence of anther. You can see here in the picture. So basically the pollen grain is formed, it has got vacuoles, it has got a nucleus, and further it breaks into two cells. One is called vegetative cell, another is called generative cell. So stages of microspore maturing into pollen grain. When the microspore matures into pollen grain, it gets two cells, vegetative cell and generative cell. Now on maturity, you can see the anther gets matured and dehiscence takes place. Thousands of pollen grains will be released into the atmosphere. And what about the structure of pollen grain? Generally it is spherical pollen grain and 25 to 50 millimeters in diameter. Cytoplasm is surrounded by plasma membrane, as you are seeing, and the pollen grain has two layers. One is exine, another is intine. So the exine layer as well as intine. Exine means outside, exo. Intine means inner layer. So once the exine and intine are formed, you can see inside there is a lot of cytoplasm. There is a nucleoli, and there is presence of cellulose in the thickening, and you can see some germ pores or pores also, germ pore. And basically it has got two cells, vegetative cell and generative cell. And what is the structure, what is the importance of exine? Exine is the hard outer layer. It is made up of sporopollenin. Again, you must remember this. This is a chemical, special chemical present in the exine, and it is highly resistant organic material. So pollen grain can withstand high temperatures, strong acids and alkaline substances. Enzymes cannot degrade sporopollenin. That is the significance of sporopollenin. Exine has apertures called germ pores, means some hole-like structures will be there. They are called germ pores where sporopollenin is absent. And there is a significance for that. Pollen grains are preserved as fossils due to presence of sporopollenin. Exine exhibits patterns and designs, as you are seeing here, and we can store these pollen grains which are very rare species of plants. We call it pollen bank. Just like how we have got blood bank, we have got pollen banks also. So very valuable, medicinally important pollen grains can be stored in the pollen bank. And further, the pollen grain has got inner layer called intine. This is the inner wall, and it is thin and continuous layer, and two chemicals are present in this intine. One is cellulose, another is pectin. And further, on maturity, a matured pollen grain contains two cells, as you are seeing, vegetative cell and generative cell. Vegetative cell is very big, as you are seeing. It has abundant food resource and a large irregularly shaped nucleus. Whereas generative cell, as the name suggests, reproductive in nature, it is small, it floats in cytoplasm of the vegetative cell, it is spindle shaped with a dense cytoplasm and nucleus. Nucleus is very predominant. And over 60% of angiosperms shed their pollen grain at two-celled stage. Okay? But in others, generative cell further divides into mitotically to give two male gametes, male gamete 1, male gamete 2. Thus pollen grains shed at three-celled stage. That means one vegetative cell and two generative cells. Generative cell 1, generative cell 2. At that stage it gets shed. So pollen grain basically shed pollen grains have to land on the stigma before they lose viability. So all the pollen grains that are dehiscensed will have to come and place themselves on stigma. So the viability period of pollen grain is variable. It depends on temperature, humidity, so many environmental factors will decide the very existence and survival of pollen grain. Viability of pollen grains of some cereals like rice, wheat is just 30 minutes. Some members of Leguminosae, Rosaceae, Solanaceae, that is brinjal tomato family, have months of viability. And what is the economic importance of these pollen grains? In what way they are useful to us? They are very rich in nutrients. Pollen tablets are used as food supplements. Pollen tablets and syrups increase performance of athletes and race horses. They are stored for years in liquid nitrogen minus 196 degrees centigrade. They are used as pollen banks in crop breeding programs. I told you just now. Pollen grains of some plants like Parthenium, Congress grass, are allergic for some people. It leads to chronic respiratory disorders such as asthma and bronchitis. So this is how finally a pollen grain is formed, and the significance of pollen grain we have understood.